Hello everyone, my name is Justin Howard. I'm presenting to you today via recording since I can't be there in person. And what I'm going to be talking about today is the logic of phylogenetics and how trees are constructed. And what I hope to cover is a good definition of phylogeny, a modern definition of phylogeny versus what was thought of prior to the early and mid 19th century. Some of the people that made discoveries that brought this field to where it's at today, how those influenced the school of thought. There was a huge wedge put in there between different camps and different philosophies when the discovery of genetic information came out. We'll talk about how it's used today and how it's utilized by researchers today, and then I'm going to cover in brief just a few different ways that these trees are analyzed and some of the options you have in doing that. So if you didn't already know how monumental of a task this is a paper I quickly found online after starting this presentation. It's published in 2019. You can see that GenBank, which is hosted on the NCBI website everybody's probably familiar with, it's a repository for genetic sequencing information. They have 450 formally sequenced species. That's out of the probably 8 or 9 million different species on planet Earth that contains bacteria, plants, and animals. So there's quite a bit of ways to go, and really we've only scratched the surface in it. So phylogeny, just as a definition, is the study of kinds, where you came from. But it's important to note that a modern definition of phylogeny also considers or also takes in that things will be related from an ancestral or evolutionary relationship. Before we knew about genetic information, it was based off of likeness. This animal has 4 legs, and so does that one, and so I'm going to group them together in their own class, and then I can continue to classify them based on other different traits. I think phylogeny is probably one of the most pervasive fields. It touches every area of our lives, from understanding how we migrated onto this continent, how archaeology, this type of pottery is going to be classified this way because they have certain type of handles versus another type. So archaeology, and then it's very useful and probably essential in ecology and conservation methods for species that may be going extinct. Helps us understand epidemiology, and obviously discerning evolutionary history, which is useful in all those things I just mentioned. So early systemists or biologists, they were limited by what they had to go on to classify things. Probably first was what they could actually observe with their eyes, and that was even dependent on how many specimens they had collected, where those, who was going to study all those specimens that were collected on voyages like Darwin's voyage he took on the Beagle. He had to get people to agree to study those specimens and help classify them. And then there was the limitation of whether they had fossils or not. They didn't always have fossils for every organism they were studying or something to compare it to. They would use categories like, based on observations, like whether there was milk produced in a living organism and its offspring, or whether it was torpedo shaped, or who knows what they thought about an octopus or a flatfish, things like that. And then their embryological development, ontogeny here, all those things factored into early classification methods. So the first person I really that's of note in this lead up to classification in the phylogeny sense, that's a modern definition, is Linnaeus. He started a taxonomic system that would be more inclusive, the higher the rank, like kingdom and domain, that would include a lot of organisms, and then as it got more specific, it would start excluding things. And this was simply a nomenclature system. He was naming things and then attempting to classify them based on their likeness with other kinds. Along came Darwin with his ideas and theories of common ancestry, descent modification, even sexual selection. That kind of threw a wrench in everything. But even he struggled with this idea of whether we should use or what we should be using to classify organisms. If you read just this excerpt here, he says, to the biologist or to the person that's trying to classify things, similar traits in organisms are pretty much useless because that doesn't say anything about their blood relationship, although it is important to the organism for survival. But these two theories, they helped answer the question or help lead people towards answering questions of how are things related in an evolutionary sense. And ultimately it led to this idea of last universal common ancestor, which flew in the face of pretty much everything that had been built up to this point. Let me go back to Linnaeus. Even before him, there was Aristotle trying to classify things, and he had his own thoughts on, just like Darwin did. Haeckel was a little bit of a character, I think. He was important in this step we're taking in the lead up to what phylogeny is today, in the fact that, or in the sense that he coined the term phylogeny with a few other terms that we still use today. I thought it was also neat that I happened to have one of his books here. He was a great artist. I never realized who he was in this sense until now, but he was originally a physician and he was German, and he had some crazy ideas about race and other things, but you can see the example of his tree here. He was building more of a ladder and not a diagram like a phylogenetic tree we think of today, with man at the top and then everything was kind of placed underneath that. Then came along Morgan, who I know I'm skipping over a lot of people that made a lot of discoveries in here, but Morgan, from my research, this is kind of the point where genetics was born. He was doing genetic experiments on fruit flies, which he won the Nobel Prize for. And one day he realized a single male with some white eyes out of the thousands he had bred with red. And he started thinking, well, how did that come about? And ultimately, he was credited with discovering that chromosomes are responsible for hereditary things being passed on through hereditary. He was also from Kentucky and attended University of Kentucky. And then finally, I've got Francis Crick and Pauling up here. There was a single paper, or they both published different papers at different times, but it was around the same time that they said, maybe we should start classifying things based on genetic sequencing and not just observations we make. And that was kind of the tipping point. Linus Pauling is probably one of my heroes. He had a ton of work. He was responsible for sickle cell anemia and understanding why that's happening in folks. And then obviously Crick and Watson made a huge contribution by illuminating how a DNA molecule was built or situated. So other ones I haven't mentioned that everybody has probably heard of, Fleming and obviously Chargaff and Mendel and the work with his peas. They all also, and many more, had contributions that led up to this point where we had to decide if we were going to keep classifying things just based on what we can see or based on things that were hidden from view, like chromosomes and DNA and genetic information. And so if you fast forward to the 90s, skipping over the beginning of protein sequencing and things like that in the 70s and the 80s, you get a group that comes together and says, okay, we're going to try to standardize how we study phylogenetics. And they are based on a philosophy. Everybody basically has their own philosophy if you think about it. They probably more questionable say that taxa or species specifically, we can't even decide what they are because things change so rapidly within individuals or within a group, and there could be such a similar appearance and code between two species that are maybe closely related geographically. There's a philosophical debate on whether we can even say there are things such as a species. Maybe we're just a collective group of organisms, or maybe that's what we're trying to study. But what is more acceptable probably is that they agree that taxonomy should be based on evolutionary relationship or kinship, and not just by those shared characteristics. Essentialism is an idea that basically everything out there in nature is already decided, how it operates is already decided, and its characteristics, and we're just left to discover it and document it. And when evolution came along, the theory of evolution came along, it went in the face of that, because now we consider that things can change, sometimes rapidly, and nothing's ever really static, and we can't just classify it and be done with it. And this opened up a big debate, a philosophical debate that is still going on today. It started, you'll see some terms called Aristotle essentialism, and they kind of started with him saying that we just need to discover creation as it is rather than creation's constantly changing. So just before we go any further, to clear up these definitions here, taxonomy is simply a nomenclature system. We can classify things with it. Nowadays, it can include morphological characteristics along with DNA, and it can be supported by phylogeny, and phylogeny can be supported by taxonomy too. The main difference is that one, in just strict sense of the definition, concerns DNA and evolutionary relationships versus the other, which does not. So getting into tree construction, this is one I've made on the website Phylogeny, and I will throw a picture of that on the screen here in a minute, hopefully. But I took 10 characters because that's all it lets you use unless you start to pay for it, and trying to get a good sampling of organisms. I've got some bacteria up here, I've got a tree, an octopus, a shark, bald eagle, a humpback whale. Down here are my mammals. And then this order of carnivorous organisms right here, which include a gray wolf, a hyena, a spotted hyena, and a lion. And it seems pretty logical that we would group them together with meat eaters. But if you notice right here, at the bottom, this is what we would call a clade, which is defined as two terminal taxa, two terminal sister taxa. The spotted hyena, which you would think is probably more canine than feline, is actually grouped with the lions. And that's because of an anatomical feature in their skull where the chamber for their auditory sensory organs is double chambered in felines, but in canines is single chambered. And so here, even today, with this modern tree and genetic sequencing included in this tree, along with the information that gave us the length of these branches here, which includes time like molecular clocks and rates of substitution in genetic code. All those things are included in this tree, and we have this peculiar, or what I would consider peculiar grouping for these three species in this clade here. One thing I don't have listed here is molecules. You know, certain organisms will be able to produce certain amino acids or molecules, and we can build phylogenetic trees off of that, as well as including it into all the data that ultimately ends up in a tree. And so this theoretical thing here, it's the more data we gather, you know, that paper I mentioned earlier from Jim Bank, the billions of nucleotides we have sequenced, the bigger the data pile grows, the more statistical testing we need, which honestly probably improves our hypothesis, because with more data we can do more testing and see which one is more valid or not. Here is the first scientific tree according to my research, that was based off of RNA sequencing. They were able to do some statistical testing, Bose and his research team, in this 1977 paper. This was published in 1977. And this looks pretty familiar to me, just thinking about what I've seen in undergrad biology classes and what I've known about this before. If I'm not mistaken, 14 right here was supposed to be for mammals. But now we're looking a little more like today in modern time phylogenetic trees. And so continuing in construction of trees, just some basic parts of it here. We've got our taxon and our species. And so this can end with species, or it can end with genus or group, depending on how specific you want to get. The branch, which if the tree you've built is meant for that, you can have this branch corresponding to the length of time, evolutionary. When I was making this tree, I could have had the option to place a number here above these branches that would be derived from the molecular clock. But it was a little bit too hard for me to discern how those numbers were being put up there and what exactly they were doing to calculate them. So I just wanted to mention it and didn't want to do an example of it. Then we've got a node, which would be a new, an ancestor gave rise to a new species at that point. And then there's the root, the last common ancestor there. We can choose to root a tree here, or we can root it in the middle of this branch, or we could rearrange this whole tree and choose to root it from the hyena down here, depending on what our research needs are. But I thought this was a good representation because it includes fossils and it includes living animals now. And we can take one of these traits, like for example the amniotic egg, and say, okay, I'm going to build a tree off of that, and all these yeses here represent this clade up here, everything with an amniotic sac in its development. Or we can take them all and integrate it all into the tree. And so now I've got 1, 2, 3, 4, 5, 6 different things, and this is one example or one hypothesis of how this tree could be constructed. Remember, no one method of analyzing or inferring the relationship in a tree is standard, but like I said earlier, every bit of data we add to the pile just helps us to make better educated guesses, especially on the theoretical side of things and statistical testing. So what can make this a difficult choice? Well, we got things like homoplasy, which I will describe here in a second. There's the time aspect and molecular clocks, which also is based off of theory. Even radiometric dating is a theory, even though it's a well-founded theory. The availability of fossils, whether we have a whole sequence for an organism or just partial. So homology would be that these species share a common ancestor. And so I've got an example here. I made this matrix in this tree. I hope it's correct. The stingers, since these three, 1, A, B, and C, since these three share that trait, we can say that they are all related with one common ancestor. And then D would not be, they would have later evolved that stinger apart from this species D. There would be an ancestor right here at this point. Homoplasy, one of the confounding factors or things that could make this difficult, let's just say that we're looking at the placenta aspect. Well, since A and B have that, it could have evolved 2 different ways in 2 different separate times. And so that would be like an example of convergent evolution, where you've got similar traits developing in 2 different species that are far removed in relationship on a phylogenetic tree. A great example of this is the octopus. It has an eye similar to human with its lens aspect. So light will enter this lens, it will be diffracted and placed onto the retina, and then we have the neurons that can form a picture for us and help us to interpret that. But if you look at this tree here that I built a long time ago for another project, I've got mammals up here and this bony fish, and all the way at the bottom is where the octopus would be at. They're far removed. They're not really related evolutionary wise. But there's so many similarities between us, it makes them a great model to study. Not just in eyes, but think of how an octopus is able to solve problems, how they're able to change their appearance rapidly, which, is something far advanced than what humans can perform. So it makes for an interesting study, and this is just one example of why convergent evolution can be studied with phylogenetic trees to help us understand development a little bit more, maybe even solve some problems. And so finally, some ways of analyzing these trees. Parsimony, which is probably my favorite so far, happens to be the most simplest by definition. And then likelihood and Bayesian are not as scary as I thought they were. And hopefully I can give you a correct definition of those. So parsimony, if we look at this placental trait here in species A and B, you can see they share it. And so the most simplest explanation would be that they developed from an ancestor that had a placenta, and it was something that happened later on the evolutionary time scale. And we can say, okay, we're going to put them into this clade since they share that trait. Going back to the matrix and the dinosaur skull picture I had earlier, we can, take that matrix and form two different hypotheses. So this one has 1, 2, 3, 4, 5, 6 evolutionary events, as opposed to hypothesis 2 here, which has 1, 2, 3, 4, 5, 6, 7. And so parsimony would dictate that hypothesis 1 is correct because it is the most simplest and it creates the environment where we would have the least amount of guessing to do. The goal is to create, in parsimony, the goal is to create a tree that prevents you from having to keep adding hypotheses to explain away what's going on in the tree branching. And so it's simpler to say that rayfish and these other ones are closely related rather than saying that a bony skeleton came about on two different occasions in two separate lineages at two different times, evolutionary wise. Likelihood is not that much worse, and I think it's well, I guess my opinion don't really matter, but I like the mode of thinking that goes into it. So depending on what parameters we want to add for our tree, say we just want to look at the genetic sequence relatedness between two organisms in a certain sequence. Well, we can take the rate of substitution that we found, or just the appearance and the frequency, or the frequency of the appearance of a certain nucleotide, put that into a computer and have it do, however many random permutations, or arrange it. We can ask that computer to arrange it in as many different ways as we want. For example, 10,000 in this graph here. And based on statistical modeling, we get a bell curve. And so we can say that this one right here in the middle is the most likely, and say, all right, I'm going to put that down for my tree determination. And then you can add other parameters to that, like the branch length, how long you think those are going to be. We can test those and make hypotheses about how long they should be, and then add that into the hypothesis of our phylogenetic tree. And you can imagine the data and the options and choices gets exponential really quick. If you look at the paper that Dr. Phillips provided to us on number of ways to arrange a tree versus taxa, it grows out of control super fast. So not only was genetic information important in determining the study of phylogeny, but computers too helped a lot. And then finally, there's the Bayesian method, which my best explanation is it's also doing hypothetical or theoretical testing from a statistical standpoint. But instead of saying this is the most common one we've found, this is the one that fell within the bell curve, we're going to say that this one is, I believe, is a certain percentage true. And an example of that would be saying, okay, tomorrow I believe there's a 2% chance of rain. And so we would look at our tree and say, okay, I believe there's a 2% chance that that tree or that that taxa is related in this manner. So hopefully I've been clear in my words, and I've been able to really talk about the logic of phylogenetic trees. It really was an exercise in logic and philosophy, more so probably than biology, I think. And I hope everybody's enjoyed this, and I wish you all the best of luck and a great rest of your week. Thank you so much.